So thanks everyone for coming. Really welcome to you all, and thank you so much for having me. I really appreciate the opportunity to present to you some of the updates from ASH from last December in multiple myeloma. That's what Dave asked me to talk about today. And thanks everyone for coming and introducing yourselves and telling me about your story. And it's really nice to meet people who have been downtown at PMH and been through the transplant process and are now back in the community and doing great. It's really nice to see. So I think you've all been sitting around long enough, so why don't we just make a start? So I'm going to try and stand here. I hope that's not in anyone's way. And we're going to talk today about things that have happened in myeloma over the last year or so. But before we start, I thought we'd start with a very brief primer on multiple myeloma, just so we make sure that everyone's on the same page. So Myeloma 101. Dave also asked me to do a little primer on clinical trials, just so that everyone is familiar with the process of drug discovery and how we go about working out as scientists and doctors how things work in medicine. Forgive me if I pitch this too high or too low. It's always very difficult speaking to a mixed audience. Some of you may be up every night reading about multiple myeloma, or other people may wait till they see your doctor in clinic and just ignore it the rest of the time. So we'll try and sort of sit somewhere in the middle. And then we're going to talk about the myeloma updates from ASH. And I think at about 1 o'clock or so, about 3 o'clock or so, we're going to have a break. And I know you've all been sitting there already in the chairs for a wee while. So just a brief overview of multiple myeloma. This is a cancer of these things here, plasma cells, usually in the bone marrow. And these cells are usually pretty useful things. They make antibodies for fighting infections, protein that circulates in the blood looking for infection. But when we have a growth of those cells, we see a spike of protein in the blood called a monoclonal spike or M spike. And these cells, when they start to invade the bone marrow, they can make the rest of the bone marrow not work so well, and the blood counts can drop. And they can also start escaping outside of the bone marrow and causing the bone destruction and fractures. And this protein that they produce, and which gets into the blood, can clog up the kidneys and cause kidney problems. And people often ask, well, how do I get myeloma? How did this happen? So in the normal immune system, there are cells called B cells that are produced in the bone marrow. That's why they're called B cells. And then they circulate around through the blood and usually end up in the lymph node. And in the lymph node, they're looking for infection. So when you get a sore throat or a cold, you often have a lymph node swell up. That's your B cell population responding to that infection. And when a B cell encounters the kind of infection that it recognizes, these cells all start to proliferate and they shuffle up their DNA to make themselves able to fight that infection even better than they originally could. And because of that DNA shuffling, sometimes when the cells go through the lymph node trying to fight infection, they make a mistake with the shuffling of their genes. And that's probably the point at which multiple myeloma starts in most people. Often 10 to 20 years probably before they actually get multiple myeloma. Those cells then normally circulate out to the bone marrow where they set up shop, and normally they produce a mixture of antibodies called IgM, IgG, IgA, a real mixture of things that when we spread them out on a gel, looks like just a smeared out hump of different proteins. But if some of those cells have a mistake, which they acquired back here when they shuffled up their DNA, they start to grow as a clone of cells, usually in the bone marrow, and they start to produce just one single type of protein, which shows up as a monoclonal spike in the blood, or an M spike. So there are really 3 different types of plasma cell disorders that we think about when we're thinking about progression from that particular situation through to active multiple myeloma. And the first of these is called monoclonal gammopathy of uncertain significance, or MGUS. This is really common. About 3% of the population over the age of 50 and 5% of the population over the age of 70 have a clonal growth of plasma cells in their bone marrow making a protein that we can detect in blood. There's about a 1% risk per year of that turning into myeloma. And over at this end of the spectrum, we have active myeloma, which is much less common than MGUS, but still not uncommon. About 1 in 150 people get it in their lifetime. And in between, we have a condition called smoldering myeloma, where if we happen to see you in clinic, we can often recognize that things have changed from MGUS into this situation, but patients still aren't experiencing any side effects, but we know they're at higher risk for going to go on and get active myeloma. For a long time, we didn't know whether all patients with myeloma had had MGUS prior, but there was a study done using banked Scandinavian blood samples in which a country in Scandinavia had banked away blood samples for a large chunk of the population over many years. And they were able to go back and look at the blood of patients who got diagnosed with myeloma. And going back 7-8 years prior to their diagnosis, 80-90% of patients had that protein present in the blood already. So all patients with myeloma likely do go through this MGUS phase many years before actually getting myeloma. So the treatment of myeloma for a long time was really limited to just a couple of drugs, melphalan and prednisone. Sorry about this. Melphalan and prednisone, which first came about in the 1960s, and then high-dose melphalan with stem cell transplant, which was developed in the late 1980s, early 1990s. And then it was really the 2000s that we saw an emergence of a large number of newer agents for treating multiple myeloma. And these are the group of thalidomide derivatives, such as lenalidomide or Revlimid, and pomalidomide or Pomalyst, as well as the proteasome inhibitors, for which bortezomib was the first one that came about in the early 2000s. And more recently, we've had carfilzomib and ixazomib, or Kyprolis and Ninlaro, which are additional proteasome inhibitors. And finally, we've had the emergence of monoclonal antibodies for treating myeloma, which we'll talk about a little bit later. So all of these drugs came about for the treatment for multiple myeloma patients as a result of clinical trials. Clinical trials is a means by which we work out which drugs work in myeloma and which drugs don't. So what are clinical trials? These are research studies that involve people. They're usually the final step in a long process of drug development that's happened for many years prior to that clinical trial ever starting. Clinical trials are responsible for nearly all the treatments we have today, apart from a few that were grandfathered in before the era of clinical trials. And in cancer therapy, clinical trials are usually designed to ask a specific question, such as new ways to prevent cancer, new ways to find and diagnose cancer, new ways to treat cancer, or new ways to manage cancer symptoms. So this is the research pipeline. Generally, it starts with discovery and basic research. So scientists may be working in the lab, or a clinician may be thinking of better ways to treat something. And there's often a lot of research that goes in at that point to really define what the question is and how it can be best be answered. And then there's some preclinical translational work that may go on between this stage and this stage. For example, a scientist may discover a drug target for multiple myeloma, and then another group of scientists may develop a drug that targets that target, and then that can advance the clinical trials. And clinical trials really occur in 3 different phases, which we'll talk about in a minute, phase 1 through to 3. And based on the results of these clinical trials, national bodies will regulate or approve that drug for use. And following approval of the drug, then there's often a negotiation period between the government or insurers for funding of that drug, and eventually it becomes available for patient care. This is a long process. So to give you some idea of how the phases of clinical trials work, of course, there's the preclinical research that often takes many years to develop, for example, a new drug. And then once that drug has been tested, usually in animal models and against cell lines in culture, then it needs to be tested for the first time in humans. And this is a phase 1 study. Phase 1 studies are really all about learning what dose is safe, what dose can be given safely in patients. And it usually involves tens of patients. Phase 2 studies are an expanded form of phase 1 studies where you're still looking for safety signatures and trying to work out how safe this drug is, but you're also starting to look and see, does it work? I'm giving it to a population of people where I'm hoping I'm going to start seeing signs that it's working. And that often involves hundreds of patients, and those studies can, phase 2 studies can run from months to years. From those phase studies, we can often identify which drugs are the best ones to take forward to phase 3 studies, ones that seem to be working and seem to be relatively safe. And so now we're narrowing down our list of drugs that we started with to a much smaller number, and those drugs into phase 3 studies. And phase 3 studies are really a test to see if this drug is better than the best alternative treatment. So if we were to give a patient a standard of care treatment at this point in their myeloma, does the new drug work better or not? And that type of study can take hundreds or thousands of patients, and often we're looking for outcome data that can take months to years to develop. So these are big studies that take a long time. And oh dear. Never going to talk with a magic mouse. Okay, and once a drug is approved after a phase 3 study, there's a lot of post-marketing surveillance that goes on for that drug. Does it continue to appear safe when it's used in clinical context by the average physician treating the average patient? Or is there a signature of new side effects emerges when we treat a lot of patients with the drug? So over the course of this process, 100 drugs that might enter phase 1 testing, we often have only about 15% of those drugs make it through to the final process of being an approved drug, and this typically costs around $100 million or so per drug. So there are several different types of trials. There are investigator-initiated trials. These are trials where a physician treating a disease might think, what if we put those two drugs together? I bet that's going to be a great combination. They're often single-center trials, or they involve only a small number of centers where a few physicians come together to do the trial. They do often take a long time to complete because of that. Funding is often difficult because often, we as physicians get paid to do our job, but we don't necessarily have funding to do trials. That's something you have to pull together using some sort of leverage somehow. And often the sample sizes are small. A slightly larger version of that is the cooperative group. So here, these are groups of physicians that all come together. They're all interested in cancer, for example. They all want to make a difference, and they all, in their center, all enroll patients on the same study that they all think is the right study to do. More like a nationwide type of study. And then there are pharmaceutical company-driven trials. So these are generally the bigger studies. The pharmaceutical companies are the ones that have invested their millions of dollars already in developing a drug that they want to take to market. They will be asking the specific question, does my drug work or not? What side effects does it have? And they will come with their bag of cash and their funding to make that trial happen. These are great trials for getting new drugs through to the market. On the other hand, pharmaceutical companies aren't often interested in asking the question, what about repurposing that old drug for treating this disease? We never thought about that before. They're not going to make any money out of that, so they're not going to fund that. And that kind of trial is going to end up in the investigator-initiated type of study. So why are those phase 3 studies important? These are the ones where patients get randomized. That means you go on the study, but you don't get guaranteed that you're going to get the new treatment. You get randomized. You get randomly assigned to either receive the standard of care treatment or the new treatment, which doesn't sound like that great a deal necessarily. Why do we need those? Well, we need these patients that don't get the experimental treatment called controls. And the reason is, the placebo effect is a big thing. It's a very real thing for both for patients and for doctors who are treating patients. We all like to think that doing something is better than not doing something. And so when we're giving a new treatment, we like to think that it's working. But that instinct is not necessarily enough. We need a scientific basis to say that when we treat a lot of patients, all with that same drug, they all do a bit better than without it. Because if we're just talking about anecdotal stories of 1 or 2 patients who got the drug and they did better or worse, it's kind of hard to separate that out from other influences that may have been happening at the time. So we need these big studies with randomized treatments. So this is an example of a clinical study design. This was the Pollock study of Daratumumab added to lenalidomide, Revlimid, and dexamethasone. And it was compared with patients that were just receiving Revlimid and dexamethasone alone. So here we start off with a set of eligibility and exclusion criteria. So they were looking for patients with relapsed myeloma who'd had at least one prior treatment and had measurable myeloma at the time that they came onto the study. And then those patients were randomized to either be in the control group receiving the standard treatment, which was Revlimid and dexamethasone, or to be in the experimental group and to receive the Daratumumab and Revlimid and dexamethasone. And in the study, there were some pre-specified endpoints. These were the things that the study doctors were going to look for as a result of, in both of those groups, and see if there was any improvement in the experimental arm over the control arm. Things like progression-free survival, or PFS, the amount of time that the myeloma was controlled before a patient started to progress and need other treatment. The overall response rate, the number of patients on either arm that responded to the treatment, the survival of patients, although this takes a lot longer to really follow, and the all-important quality of life. Was this something that people could tolerate? So when participating in a clinical trial, the advantages are that you may get access to new treatments that aren't available to the public. Your average doctor can't go and prescribe this medicine. It's not available over the counter. The new treatment may be more effective and may have less side effects than the standard of care treatment, possibly. As a result of being on a clinical trial, you may end up being treated at a leading cancer care institution where people are very engaged with this disease and with clinical trials and new treatments. You may help others by furthering the understanding of myeloma and its treatment. And I think that's important. And if you're involved in a trial, you may get some of your costs reimbursed, such as parking, which downtown, believe me, is really important. So on the downside, the treatment may not work, or it may have unexpected side effects. There's definitely going to be more tests and visits involved with being on a clinical trial and not being on a clinical trial. And if you go on one of these phase 3 studies and are randomized, you may not get the experimental treatment. You may end up getting the standard of care treatment and yet still have to do the more visits and tests. And the new treatment may not be immediately available at the end of the study, even though you've gone on the study or on the control arm. So how do you get on a clinical trial? Well, you should ask your doctor if you think you're right. Usually, most clinical trials are looking to enroll patients at some particular point of time in their myeloma, so either at diagnosis or when they're changing treatment because the myeloma is progressing. Those are the usual time points that people get enrolled. Often clinical trials, because they're looking for specific subgroups of patients, they're going to have very particular eligibility criteria. So if you think that there might be a trial that you're interested in, the next thing to do is to look at the eligibility criteria and the exclusion criteria. And you can have your doctor or the trial coordinator talk to you about the study and give you an overview about that. And then if patients look like they might be eligible for the study, they're given a consent form which is discussed with them, but they're not allowed to sign it the same day. You have to take that away and read it at home, think about it, and return it if you're happy to proceed. So questions to ask if you're interested in a trial. What are the details of the treatment? Is the treatment oral or is it an injection? And if it's an infusion, how long does that last? How many visits does the trial involve and how frequent are they? And the duration of the treatment, is it ongoing or is it just a few months? Will you always get the treatment, or is the study a randomized one in which you may not end up with the study treatment? And if it is randomized, is there crossover? So if you end up in the control arm getting the standard treatment, at the end of that time or when you need it, are you able to cross over and get the study treatment? Which sometimes you can. And will you know whether you're getting the experimental treatment or not? Because a lot of these studies are blinded, and so you don't know whether or not you're getting the experimental treatment. And are extra tests needed? A lot of the studies in multiple myeloma do require extra bone marrow biopsies, so you should know that going in ahead of time, and often CTs or X-rays or blood tests. And what are the expected side effects or potentially serious side effects? And are there extra costs to you to come in and be involved on a study? So this is just an overview of the clinical trial process. Usually there's some executive guy in a drug company who's happy with a plan that his scientists presented to him for studying their new drug they've just developed. And so he approves a protocol that for doing a study. And then the pharma company goes out and looks for doctors that they want to help do the study. And then once these sort of negotiations are made, then doctors seek approval from the institutions to run the study at their site. And once the ethics boards and the lawyers at the institution have looked at the study and approved it, then patients are allowed to start being recruited into the trial, and they're followed, they're given the treatment, all of that data eventually gets entered into a database, and then some statisticians eventually end up looking at that database, working out whether the drug works or not, and the results get presented to peers, so it gets peer reviewed by other doctors who are not involved in the study. Is this right? Did they come to the right conclusion? And it also gets reviewed by regulatory authorities like Health Canada or the FDA. And finally, if the drug meets all the requirements, it gets approved and registered, and then patients can receive it. And this whole process takes years. But it does work, and it's led to some amazing advances in myeloma. So now we're going to start talking about some of the updates in myeloma, which is probably why you're all here. And the first thing I want to talk about is Daratumumab, for the very good reason that this is now funded in Ontario. So exciting news, and I think we should look a little bit about the trials into Daratumumab and how we want to use it now that it's funded. So Daratumumab is one of these things called a monoclonal antibody, and these antibodies stick onto the outside of tumor cells, and then they do complicated things. They draw in a number of other immune cells that can now recognize the tumor cell or the myeloma cell and destroy it. So these are really ways of enhancing your immune response to the tumor cell. Now these antibodies are not like drugs that are made by chemistry. They're actually proteins. A little bit, if you think of egg white, that's protein. So these things are made by cells, usually. The cells are engineered to make these proteins that then can recognize myeloma and draw in the immune system to attack the myeloma. And the big news was that just 3 weeks ago or so, Cancer Care Ontario finally announced funding for Daratumumab. So this is now funded in Ontario, but only under a certain particular restrictions at present. So it has to be funded either with Revlimid and dexamethasone or with Velcade and dexamethasone. And patients have to be eligible to get the funding for those drugs before they can be eligible for getting the funding for the Daratumumab. So the reason that Daratumumab was funded was based on 2 studies that were published in the New England Journal. The first of these was the CASTOR study, which was testing the addition of Daratumumab to Velcade and dexamethasone in patients who had relapsed myeloma. So in this particular study, nearly 500 patients with relapsed myeloma and who had had one prior treatment and had active myeloma or measurable myeloma were randomized to receive either bortezomib and dexamethasone or Daratumumab and bortezomib and dexamethasone. Just go back here. And for the first 8 cycles, patients got the bortezomib and dexamethasone, but after 8 cycles, they actually stopped the bortezomib or the Velcade and dexamethasone. And in the treatment arm, the experimental arm, the patients continued to get Daratumumab after that, but in the control arm, actually patients weren't receiving anything after 8 cycles of treatment. And amongst the patients who went on to the study, most of them had a median of 2 lines of treatment. and the majority had received prior Velcade, prior Revlimid, and about half of them had received both of those before. And so this was the result of that study. And here you can see the proportion of patients that remain free of myeloma after going on to treatment. And as these curves drop down, that means increasing numbers of patients are progressing. And this is time on the bottom axis in months. And so you can see that the addition of Daratumumab meant that more patients were doing better for longer and having less progression of their myeloma. And in fact, the addition of Daratumumab to Velcade and dexamethasone for relapsed myeloma improved the median progression-free survival, and there was a 61% reduction in the risk of progression when you add Daratumumab to Velcade. So the other study was the Pollux study, and it was done in parallel. And it was essentially the same question, but asking whether addition of Daratumumab to Revlimid and dexamethasone this time was better than Revlimid and dexamethasone alone, also for patients with relapsed myeloma. And so in this study, more than 500 patients with relapsed myeloma who had had at least one prior treatment and had measurable myeloma were randomized to receive either Revlimid and dexamethasone every 28 days, like most people get it, as long as their myeloma continued to respond until progression. Or they were randomized to receive that same treatment plus Daratumumab, given on the usual Daratumumab schedule, which is weekly for cycles 1 to 2, and then every 2 weeks for cycles 3 to 6, so that lasts 16 weeks, and then every month until progression. And these are results of the Pollux study. And once again, you can see that the addition of Daratumumab to Revlimid and dexamethasone meant that a much larger number of patients were staying in remission for longer than patients treated with Revlimid and dexamethasone alone. And we talk about a hazard ratio. Progression is the hazard, and the hazard ratio here is 0.37, meaning that more than 60%, there was a more than 60% reduction in risk for patients receiving the Daratumumab together with Revlimid and dexamethasone. So these are very, very good results, much better than we see in a lot of clinical trials, and this made it really easy to advocate that we should have Daratumumab in Canada to treat patients. It looks like patients on Daratumumab Rev-Dex are going to, on median, are going to progress out here at like 40 months compared to about 17 and a half, 18 months with Rev-Dex alone. So addition of Daratumumab to Revlimid and dexamethasone for relapsed myeloma significantly improved median progression-free survival with a 63% reduction in the risk of progression. This is a complicated table. I don't expect you to take it all away, but these are all different subgroups of myeloma patients with different treatments, previous treatments, number of lines of treatments, whether they have high risk or standard risk cytogenetics. And all of these patients are doing better on the left of one, are doing better when they receive Daratumumab than when they didn't receive Daratumumab. You notice that high-risk cytogenetics, there is a bit of a crossover of this line here, so we can't prove that high-risk cytogenetics patients are doing quite as well as the other patients. If we look at the side effects of Daratumumab, they're generally pretty reasonable, actually, compared to a lot of treatments that we see coming through the clinic. So in green is the side effects that you get just with Velcade dexamethasone alone, and in red is the 3 drugs together. And for most of the side effects, they're relatively comparable between the 2 arms. What you will notice is that Daratumumab, of course, can cause infusion-related reactions, and you don't get that in the control arm where there is no Daratumumab being given. And the reason that patients get this is because Daratumumab is a protein, like an egg white. And so just like people can have egg white allergies, you can have a bit of a reaction when you get a protein infusion. And it's very common with all monoclonal antibody infusions that people can have a bit of a reaction. It's often a bit of a fever or maybe a blood pressure change or some hives or some coughing and some shortness of breath. It usually just happens when the infusion is happening. Often it's only the first time you get the infusion and not subsequent infusions. And usually the nurse just turns down the rate for a bit and then turns the rate back up again. And might give you an antihistamine or something. Daratumumab also does increase the rate at which patients get low white cell counts. In this particular study, there was a little bit more diarrhea and there's a slightly increased risk for infection, though it's only a small increased risk. In the Velcade study, they actually broke down the benefit of adding daratumumab according to whether you were getting it after 1 prior line of treatment here, after 2 prior lines of treatment, or after 3 or more prior lines of treatment. And you can see that when you add daratumumab to Velcade and dexamethasone, it really gives you the biggest bang for buck, or the biggest benefit when you're doing it after 1 prior line for treatment in the relapsed setting. So it's better to use early on rather than later in the course of myeloma. And that same data here is shown here, that after 1 prior line, 2 prior lines, or 3 prior lines, you get more benefit out here after 1 prior line than after 2 or 3. Also, if we look at the ISS stage for myeloma, the amount of myeloma that you have, it works better for lower amounts of myeloma than for big amounts of myeloma. So here's a quick summary related to daratumumab when it's used for relapsed myeloma. So in relapsed myeloma, the addition of daratumumab to either Revlimid and dexamethasone or to Velcade and dexamethasone reduces the progression risk by a factor of about 0.37, 0.39, something like that, quite a lot. That's reducing that risk by quite a lot. It improves the median response duration, or the projected progression-free survival of patients on Revlimid dexamethasone by roughly 22 months when you add in the daratumumab. That's an average for all of the patients that received 1 prior line, 2 prior lines, 3 prior lines. And it improves the median response duration, or the PFS, for Velcade and dexamethasone by roughly 20 months, particularly for patients with 1 prior line. But that 20 months does drop off, more so with Velcade and dexamethasone for patients that have had more than 1 prior line. So if you're going to use it with Velcade and dexamethasone, it's better to use it after only 1 prior line, but if you're going to use it with Revlimid and dexamethasone, it does appear that you can use it after 1, 2, or 3 prior lines. So what about the addition of daratumumab to frontline therapy? If it looks like it's working better after 1 prior line than after 3 prior lines, what about after no prior lines? What about if we used it for newly diagnosed myeloma? That was the obvious next question. This is a summary of some of the trials that have been done with frontline treatments in patients that can't receive transplants, who are transplant ineligible. So this is their frontline therapy and the trials that have looked at that. And the first thing to notice is that some of you may have had VMP way back. This used to be our standard of care after it was shown that it gave 18 months progression-free survival in myeloma, which was good back then. But since that time, there was the first study that showed that Revlimid and dexamethasone, when used as frontline treatment for myeloma, actually gave better frontline responses and kept the myeloma at bay for more than 2 years, 25 months on average. And that was the first study. And since that study came out, Revlimid and dexamethasone has been funded as frontline treatment in Ontario for transplant ineligible patients. But if we look down here, this was reported, not the most recent ASH, but the ASH prior. The addition of daratumumab to VMP actually changes the situation, so that now when you add daratumumab to VMP, patients were not even progressing on median at 30 months, which is better than the Revlimid and dexamethasone of 25 months. So the addition of daratumumab up front, of course, was making a difference as expected. But the question then became, what about when you add daratumumab to Revlimid and dexamethasone? If Revlimid and dexamethasone are the better backbone, then as they were up here, what about when you add daratumumab to that? And that's just been reported out at ASH, that study. So this is the study. The addition of daratumumab to Revlimid and dexamethasone, the MAIA study. This is the study design. So patients had to have transplant ineligible, newly diagnosed myeloma. They had to have a good performance status, meaning they weren't too sick, and they had to have reasonable kidney function. And then they were randomized 1-to-1, meaning one patient went here, one patient went here, to either the Revlimid and dexamethasone frontline treatment or to the daratumumab and Revlimid and dexamethasone frontline treatment. Oh, sorry, my apologies. And following that randomization, patients were treated until progression, and the outcome that the trial investigators were most interested in was looking at how long it kept their myeloma away, how long it kept their myeloma controlled. as well as a number of other things, including safety, survival, and the response rate. And so this is the outcome data, or the efficacy data, showing that with the addition of daratumumab to Revlimid and dexamethasone, after more than 30 months of follow-up, nearly 3/4, more than 2/3 of patients are still in remission, compared with the Revlimid-only arm, which is approaching median progression there at just over 30 months, which was comparable to the previous first study. And if you project this line out, you can see that the median progression-free survival of patients, the median time that patients' myeloma is going to be controlled using frontline Dara, Revlimid, dexamethasone, is going to be more than 4 years. So you're going to get diagnosed with myeloma, maybe told that you're not transplant eligible, but you can go on this frontline treatment that's going to keep your myeloma in remission on average for 4.5 years. So that's quite an improvement from where we were at a few years ago. The hazard ratio here is 0.56, so a very good improvement over even what was quite a good treatment here. So 44% reduction in the risk of the myeloma progressing in newly diagnosed patients. This compares with the Alcyon data, so that's the Dara VMP study that had been reported at the previous ASH. This is the updated data. Again, you can see that the addition of daratumumab to VMP also works well, also with a very good hazard ratio. But if you look at the absolute numbers, this is 30 months here with Dara VMP, 60%, and here's 30 months here with Dara RD, 71%. So frontline therapy, it does look like the addition of Dara to RD is going to be better than the addition of Dara to VMP. And so I think Dara RD is going to become one of the standards of care eventually. It's not yet funded that way in Ontario. This was only just reported out at ASH, and these funding things take a while, but certainly that's very encouraging data. And if just to look at the efficacy across all different patient groups, so male or female, actually female are doing slightly better, younger and older, by race, by stage. In all groups, the addition of daratumumab to Revlimid and dexamethasone was beneficial. Interestingly, there was a particular benefit if you didn't have IgG myeloma for some reason. And if we look at high-risk cytogenetics, again, the addition of daratumumab not quite doing quite as well as we would like. It's not having as much benefit as we would have hoped over and above the standard RevDex. So the bottom line is that daratumumab, Revlimid, dexamethasone significantly reduces the risk of progression or death across the majority of the subgroups. So here's a quick summary for newly diagnosed myeloma and the role of daratumumab. This was the updated finding from ASH. So newly diagnosed myeloma patients, the addition of daratumumab to either Revlimid and dexamethasone or to VMP, Velcade, melphalan, prednisone, reduces progression risk by a factor of about 0.43 to 0.56, and it improves the median PFS, that's the response duration, by roughly 21 months in both arms when used frontline, in both treatments, VMP and RD. And Dara RD is predicted to provide progression-free myeloma control for more than 4 years in 50% of newly diagnosed patients. So daratumumab works, this is the take-home message, I guess. It's hard to remember all those numbers and things, but the take-home message is daratumumab works well when combined either with Revlimid or Velcade regimens, and sometimes doubles the response duration to either of those drugs alone. It works at its very best in newly diagnosed patients or at first relapse. However, even after 2 to 3 prior lines, addition of daratumumab to Revlimid and dexamethasone gives long responses if you're not already refractory or resistant to Revlimid. And still, even after those number of lines, use of daratumumab added to Velcade and dexamethasone is still better than Velcade and dexamethasone alone, but it's not the optimal usage of daratumumab. So your doctor should be really, or you should be considering, or your doctor should be considering how you can get daratumumab. Should you begin it now or with your next treatment? Because a lot of myeloma care revolves around strategizing how to use the drugs that are available and funded. And daratumumab is now funded with Revlimid or with Velcade, and you need to use it when you get one of those drugs. And if you're getting one of those drugs for relapsed myeloma, you should get daratumumab with it. If you're on Revlimid and dexamethasone now, you can actually have daratumumab added. If you're on Cyber-D or something for relapsed myeloma, you can drop the cyclophosphamide and add daratumumab, which would be a great strategy. So you should talk to your doctor about that, because if you don't do it now, then when? You know, if you've had Velcade or if you were on Revlimid maintenance and then you've progressed on Revlimid maintenance, now you're resistant to Revlimid, you're not allowed to get Dara-RD, so you're going to have to get Dara with Velcade. And so the next regimen should probably be Dara Velcade Dex, or it should be high on the list anyway. You need to discuss it with your doctor.
You mentioned this approval is for Daratumumab and Revlimid and Velcade if you're eligible for Revlimid and Velcade. And if you've taken either one of those before, are you ineligible, or is there some way around that? Because
right, so the question is, if you've had Revlimid and Velcade before, are you ineligible to get funding for those, and does that mean you're not going to get daratumumab? You have to be able to get one of those drugs funded to be able to get the daratumumab. It has to be given with one of those drugs. So often though, if you've had the Velcade before, for example, if you had it before transplant, you're eligible to get Velcade again later on, because you're not resistant to it. You've just had a course of treatment, a limited course of treatment with it. You can get it again later on, and you can get the daratumumab with it. And I think that's going to be the case for a lot of our transplant patients, is they got Cyber-D, then they got transplant, and then they got Rev maintenance, and now when they progress on the Revlimid maintenance, they're actually resistant to the Revlimid, so they can't get Dara-RD. So they're going to need to get the daratumumab with the Velcade, and so the next line would probably be Dara Velcade Dex based on this current treatment. Now the other caveat to the funding in Ontario for daratumumab is that you can only get one expensive triplet funded by the province, which means either you get Dara-RD or Dara-VD, or you get KRD, because those are, I guess, the expensive triplets, the carfilzomib Rev Dex. So you'd have to make your choice as to whether you went, to get carfilzomib, Revlimid, dexamethasone, or daratumumab, and one of, and Revlimid or Velcade in combination. And if you're actually on carfilzomib and Revlimid and dexamethasone at present, you have until mid-June to switch over if you prefer to switch over.
Is this only after the first relapse has occurred that you would take this drug?
No, you can use it later. You can, as long as you're sorry, the question is, would you only use the daratumumab after a first relapse, or can you use it later?
Or no, before.
Or before?
Before you relapse.
It's not funded for newly diagnosed patients in Ontario yet, but the data are just coming out.
It's a post-stem cell before relapse.
It's also not funded for maintenance. Yeah, there are studies ongoing, and I suspect it'll be really useful, but it's those studies haven't reported out, and there's definitely no funding for that yet. And again, this is a drug that costs a lot of money, more than $100,000 a year, just like a lot of all the other new drugs. And when you use them in combination, you're talking about a lot of money, and the province is pretty clear about when you can use it and what combination.
I know I needed this help after my first transplant last year. I'm not taking stuff for that gas. Do I assume that it does not have, if I relapse, they wouldn't have the error to it now as long as I'm not relapsed?
If you're on Rev Dex, your doctor may have got that for relapsed myeloma, because you have to take dexamethasone when you get the Revlimid for relapse. Whereas most patients who don't take dexamethasone, who take Revlimid without dexamethasone, got it funded as maintenance. So there's two different funding routes for Revlimid. If you're taking the dexamethasone with the Revlimid, your doctor may have got the Revlimid by applying for it for relapsed myeloma. And yes, you can add daratumumab to it now, and you probably should.
I just realized before I had the third transplant, I relapsed and I went on to Rev Dex. But then I had the transplant last summer, and now I'm on Rev Dex.
So that got funded actually for relapsed myeloma. We have to play these, we have to work the system as much as we can. So you can add daratumumab to that. Yeah. So just, I guess we're going to do a question and answer. Oh, another question at the back.
In a situation where I may have to find another medication to supplement, in this case, to replace it regularly, in the sense that I've been going through a series of them and the things are not going very steadily. I started off with melphalan and the prednisone for my autologous stem cell transplant. And then, so that sustained me for 8 years. And then after that, I started losing strength. I was given Barricade and Dex, but that combination was very bad on me. And now, since then I've had the Revlimid and seems to be doing quite well. But I'm worried that if it fails also, then I want to really have to else.
So it sounds like, so I didn't quite catch it all, but it sounds like you're getting Revlimid for relapsed myeloma, because you had a relapse, you got the Velcade Dex, and then Revlimid. So even though you're doing well, you could do even better by adding Daratumumab and keep that remission, make that remission last longer. If you already used up the Velcade for relapsed myeloma, you probably want to think about adding Daratumumab now, although that's something you need to discuss, with your doctor, because there may be reasons why this isn't a good thing for particular people, but for most people that's going to be the right choice. Okay, so just a very brief update on maintenance treatment after transplant. So as you know, most people get Revlimid maintenance after transplant. And this is based on 2 studies that showed a very good improvement in progression-free survival, the amount of time the myeloma remains in remission after transplant. When you add 1 dose of Revlimid a day or 1 dose of Revlimid for 3 weeks out of every 4 weeks. So at ASH in 2018, or a few months ago, there was a report on the tourmaline MM3 study. So this is a study of the use of a proteasome inhibitor, an oral proteasome inhibitor called Ninlaro, as maintenance after transplant. So this is, you can think of this as an oral version of Velcade, given as 1 tablet once a week for 3 out of 4 weeks. So basically 3 tablets a month, given as maintenance treatment for myeloma. And in this study, patients, newly diagnosed patients underwent standard of care treatment for the myeloma, whatever that was locally, and then they were transplanted. And they had to have received the transplant within 12 months of starting treatment, and they had to have responded to treatment during this time. And then they were randomized to receive either Ixazomib or Ninlaro, 1 tablet a week for cycles 1 to 4 at a dose of 3 milligrams, and then increasing to the full dose of 4 milligrams for 3 tablets every month thereafter, or placebo. So it was a randomized study, and patients didn't know what they were getting. And this is the result of the study. So again, the use of tourmaline, which is use of oral version of Velcade, Ninlaro, Ixazomib, reduced the risk of progression by about 39%. So encouraging that it did do this, and it's clear that it's a useful drug. However, it is also true that it's not quite as active, at least in controlling progression, as Revlimid was in the studies that looked at Revlimid maintenance. So, but there may be particular reasons why we choose to use Ixazomib over a Revlimid. For one, due to side effects that a patient may have, or due to this little fact here that the high-risk patients were responding quite clearly from the Ixazomib as much as standard risks. They had a good improvement in their hazard ratio matching a standard risk patient. So they weren't being left behind in terms of improvement in risk. Also, it was older patients were doing well with it, and also patients with a big burden of myeloma, ISS stage 3. So it's a useful drug that can be given as maintenance treatment after transplant. And some patients get a second transplant, or even, I believe there was one here who had a 3rd transplant. And Revlimid's only funded for maintenance after 1 transplant. So if you need maintenance after another transplant, here's an option. It was generally well tolerated, but there was an increased risk for nausea and diarrhea. Some patients even had some vomiting, at least at one point during the course of their treatment. There's a more drop in platelet counts when patients receive this kind of treatment. So it's not without side effects, but it was generally well tolerated. And the all-important quality of life was really overlapping with patients who were getting placebo. treatment. So patients themselves judged that the quality of life was pretty good. This is on a scale, 0 to something, 100 or 90 in patient. This is 70 here. So the next part of the talk is about CAR T-cells. This is something that comes up a lot in clinic. I know there's a lot of patients that are interested. It makes the news a lot, so it's natural that people would see it in the news and want to know about it. And there's a lot happening at ASH about CAR T-cells, so I thought I'd present a section on this. This is all still investigational and at a research level, but this is something that could be on the horizon, so I think it's important to take a little time to look at it. So CAR T-cells are these little T-cells here, which are engineered, redirected to attack tumor cells. And when tumors develop, like myeloma, they usually learn ways to escape the immune system. That's how they develop. If they couldn't do that, they wouldn't develop. So we have to re-educate the T-cell to be able to recognize the myeloma cell and attack it and destroy it. And so one way that that can be achieved is to genetically engineer some T-cells with something called a chimeric antigen receptor, which is basically a protein that's been designed to target something on the tumor cell, together with some other pieces of protein that stimulate the T-cell when it recognizes a tumor cell. And so these chimeric antigen receptor T-cells are genetically engineered to recognize tumor cells, and then they're essentially augment the normal T-cell response against tumor cells. So this is how it works in practice. A patient has to first give up some T-cells in a process called leukapheresis, which is like doing a stem cell collection for those of you who have had transplant. But in this particular case, the goal is to collect T-cells from the patient. The T-cells are purified, and then this chimeric antigen receptor is introduced genetically into the T-cells, which are then grown up in tissue culture and expanded to large numbers of cells. They're purified, all of the reagents are removed, and so you're just left with the chimeric antigen receptor cells, the CAR T-cells. Then the patient's actually given some chemotherapy to dampen down their normal T-cells to make way for the new CAR T-cells that are going to be reintroduced. And then the CAR T-cells are given back in an infusion. Now, there's been a number of different generations of CAR T-cells. In fact, these things have been around ever since the 1980s. I remember hearing about them back then, and they didn't really create much of a ruckus back then because they didn't work. And it's taken 20 years of development to really get something that's now very effective. And so the important parts here are that there's a part of the CAR T-cell that recognizes the tumor, and it has to be engineered specifically for each different type of tumor. And sometimes not all patients with the same type of tumor are going to respond to the same type of CAR T-cell. And then there are some stimulatory motifs in the receptor tail that's inside the cell that stimulates the T-cell when it recognizes a tumor cell. And getting the right combination of these things is what has taken us 20 or 30 years. And so at ASH this year, there were a number of different talks on CAR T-cells for multiple myeloma. This is just some of them, the ones that I managed to capture. I'm really only going to present one of these because most of these studies are all very early stage, and most of them are giving the same kind of results. So this is the Bluebird 2121 CAR T-cell. It's actually owned by Celgene at present, and this was the CRB 401 phase 1 study that we have open, or we had a similar version of this study open downtown at Princess Margaret, although we had difficulties recruiting a patient at the right time onto the study when there was a study spot. But nevertheless, so in this particular study, patients were leukapheresed, they had their T-cells taken, and the CAR T-cells were manufactured. And meanwhile, 5 days before the patients were given the CAR T-cells, they started the chemotherapy, then they were given the CAR T-cells, then they had some bone marrow biopsies at week 2 and week 4, and their response was assessed at week 4. And different patients were given escalating doses of the CAR T-cells all the way from 50, I think that's 50 million, all the way up to 800 million. And then after these patients had had their CAR T-cells, there was a dose escalation a dose expansion phase where more patients, 22 more patients, were also treated in the dose range of 150 to 450, which was found to be the active dose range for the CAR T-cells. And so patients going on to this study had a median of 7 or 8 prior lines of treatment. So they'd had everything that was available to treat myeloma. They were, I guess, had run out of other options. And 100% of them had had prior transplant during the escalation phase. And many of them had, large numbers of them had seen all these other drugs: bortezomib, carfilzomib, Revlimid, pomalidomide, Daratumumab. Not all of them Daratumumab. And so this was the response rate. So the response rate was proportional to the dose of CAR T-cells that were given. So more patients needed more CAR T-cells. You couldn't use the lower dose. You had to use at least 150. But when you were using 150 million cells, the response rate was up here at 95%, with half of patients going into a very deep response, a stringent complete response or a complete response. So, for patients who've had everything and then the response rate's 95% and half of them, you can't detect the myeloma anymore, that's a pretty striking result. And even though in this study they only allowed patients onto the study if they had this BCMA protein that was expressed on the myeloma, which most myelomas do, but they required that patients have at least half of their tumor cells expressing that at a reasonable level to get on the study. They found out that the response rate was relatively comparable between patients who had high levels of that protein and patients who had low levels of that protein. Although the patients that had high level of that protein, which is actually the target for the CAR T-cells, had deeper responses. But I think one of the drawbacks or one of the slight disappointments with the approach so far is that the responses aren't that long-lived yet. So for the group overall who got the low dose of CAR T-cells, the response was less than 3 months. For the patients that got the active doses of CAR T-cells, the response was around a year or so on average. And even amongst the group that got the really deep responses where the myeloma wasn't detectable anymore after the CAR T-cells, the response was just under 18 months on average until the myeloma came back. So this is a group of patients who've had everything else, so it's a heavily pretreated population. But given the deep responses, it would have been nicer if we'd seen longer responses with this kind of treatment. So part of the problem still with the treatment, I think, is the persistence of these T cells. It's very difficult to engineer T cells and get them activated in the body and then to hang around. Because T cells, once they're activated, they tend to be activated for a while and then get exhausted, and then their numbers plummet and they don't hang around. And that's exactly what we're still finding with the CAR T cells for myeloma, is that, even with the higher doses of CAR T cells, this is the 800, this is the 450 million dose, the CAR T cells are dropping back to very, very low levels by 6 to 12 months. And because these CAR T cells are probably not getting rid of every single myeloma cell, you need the CAR T cells to hang around to keep fighting the myeloma. And when the CAR T cells don't survive for long enough, then the myeloma comes back. So one funny slide, but one particular problem with CAR T cells are the early side effects. And there are 2 particular early side effects that we have to watch out for, which are cytokine release syndrome. This is when the CAR T cells, they get reintroduced back into the body, they're engineered to recognize myeloma, and all of a sudden they see a lot of myeloma and they become really activated, and they release lots of hormones called cytokines, which basically the things that give you fever and drop your blood pressure when you're sick with an infection. And these CAR T cells make a lot of those cytokines very suddenly, usually within a few days or a few weeks of the initial infusion. And that can be quite dangerous if you get that in a bad way. So patients have to be monitored very closely during the initial few days and weeks after their CAR T cells. They may need special monoclonal antibodies against cytokines to prevent them becoming really sick. So it's not a easy treatment to necessarily give. The other thing is that occasionally patients can get neurological complications with brain swelling. And so again, that needs to be monitored closely, and patients may need steroids to treat that. And blood counts do tend to drop quite a bit after CAR T-cell therapy, so there's a risk for infection too. So the take-home message for CAR T-cells at the moment in myeloma is that they're promising, but they're still experimental and they're still at an early stage. They can produce high response rates and deep responses, but the duration of the CAR T-cell persistence and the duration of the response is really not as good as we would like it yet. And there's this association with early side effects that needs to be really carefully managed, and make it not an easy treatment to give. It needs to be done in a specialist center. And the current cost is around $500,000 per patient, and with the median response of 12 months, it's going to be hard to fund it at this current level of efficacy. So it's still something that's actively being researched and watched the space, but I think we're going to need probably a few more years to really get to this being an actual treatment for myeloma that we can prescribe. Having said that, in acute leukemia, ALL, and lymphoma, it's getting to the point where this is a treatment that we can prescribe. There's a tail of patients who appear to be cured or living a long time with that treatment. The caveat there is the effectiveness of this treatment all comes down to the target that's being targeted on the tumor cell. And I don't think BCMA, although it's good, is as good as the target for acute leukemia. And so that means that the CAR T cells are not working as well in myeloma yet as they are in acute leukemia or lymphoma. So I'm going to talk now about a slight twist on the approach of using CAR T cells, and that's what I believe to be an equally exciting, if not potentially more exciting, approach of using bispecific T-cell engaging antibodies, or BiTEs. So these are antibodies. They're a little bit, remember Daratumumab, is a monoclonal antibody. These are not monoclonal antibodies, but these are types of antibodies that are specifically engineered to bind both to the tumor cell but also to the T cell and bring the two together and activate the T cells that are already in the body. So you don't need to take the T cells out of the body and engineer them. You just throw in this BiTE, like you give a drug, and it brings the T cells and the tumor cells together and activates the tumor cell, activates the T cell against the tumor cell. And there are a number of different targets that are being tested on the myeloma cell in myeloma for these bispecific T-cell engaging molecules. Time will tell which one of these is the best one. This is BCMA, which is the one that's being targeted by most of the CAR T cells. You can see that the myeloma cells have a lot of this on their surface, whereas normal cells in the bone marrow don't have any, so that allows the T cells to home in and target the myeloma cells. So at ASH this past year, there was the first report of using a bispecific T-cell engaging molecule in myeloma, and this was an Amgen product. So they had taken an antibody that recognizes something on T cells, an antibody that recognizes something on myeloma, recognizes the BCMA, and then they linked the two together molecularly, and they developed this thing called a BiTE, or AMG 420 is its current name. And then this works by infusing it into patients, and then it binds to the tumor cell and it binds to the T cell, brings them together. As you can see in this picture, this is under the microscope. Here's the T cell attacking the tumor cell. So in this particular clinical study, it's a phase 1 study, which involved 6-week cycles of treatment, and within each 6 weeks, patients were treated with a 4-week continuous infusion of this drug. This drug has a very short half-life, so unfortunately patients had to carry around a pump and have it infused continuously for 4 weeks out of every 6 weeks, and patients were allowed to be treated for up to 10 cycles. And this was really a dose-finding study and a safety study for this first time that this type of drug has been tested in myeloma. There were a few significant side effects, and one of them was infection. There were a number of patients that had serious infections, and whether that's because their myeloma was quite advanced and they'd had a lot of other treatments before, or whether it's because they had a needle in their arm or somewhere, and for 6 weeks at a time. And so that was increasing the risk for infection. But there were a lot of infections, and 1 or 2 patients also got some significant neuropathy, which was unexpected. These were the responses. So they started at quite a low dose of the drug in yellow, and they worked all the way up to the blue and purple doses. And you can see that at the blue and purple doses, most patients were responding, whereas at the lower doses, very lower doses, the majority of patients weren't responding, though some people still responded even at the very low dose of treatment. And this created a lot of waves in the media and investment fields, because this is another new treatment that looks exciting on the horizon, I think, for myeloma, although it still needs some work. So the reason for discontinuing most patients, although those initial results were very encouraging, most patients have actually since come off the drug, either because their myeloma had progressed or because of infections or other problems. Of the patients that did respond, there were, again, like with the CAR T-cells, there were a large number of patients that became MRD, or minimal residual disease negative, with a stringent complete response, meaning that their myeloma was undetectable by all our current means at present. So there were a number of patients that did get very deep responses. But again, these patients are relapsing even though we can't detect them with all our current standard methods for detecting minimal residual disease, which tells us something about our methods for detecting minimal residual disease, I think. So we do have a trial opening for this agent, or actually I think it's the derivative agent that has a longer half-life and can be given by injection rather than by continuous infusion. So we also have another of these types of molecules open in clinical trial at PMH, and I can't show you any human data because nothing's been reported yet, but I can say that in mouse models and in primate models, this drug works really, really well. It gets rid of plasma cells all the way, and B cells, so it gets rid of the whole lineage of cells that might be involved in generating myeloma. So this is this molecule here. It's got this great name, BFCR 4350A from Genentech. Genentech's a good company. I like their product, and I like this one, so I think this is going to be a promising new agent. I have a patient on it who just had a great response first cycle. So another class of new drugs in myeloma are the antibody drug conjugates, or ADCs. These are monoclonal antibodies that have been designed to bind to a tumor cell, but instead of relying on the immune system to then do the work of getting rid of that tumor cell, they actually carry some sort of drug that's going to poison the tumor cell. So they're an antibody drug conjugate. And there is a very good antibody drug conjugate that's under clinical trials for multiple myeloma at present. A lot of the work was actually done at Princess Margaret Hospital and was led by Suzanne Trudell. So she presented this work both years at ASH, and it's just recently been published. So this is an antibody drug conjugate that actually targets BCMA, just like all the CAR T-cells are doing. So they're all after the same target on the surface of myeloma cells. And then it's linked to a certain type of plant poison that's good for killing tumor cells. And in the patients that were treated, you can see that the majority of them had responses. This is their myeloma protein, and most patients who were given the drug responded, particularly when they were given higher doses. And for a number of patients, this was quite a long-lasting response. And so this is the progression-free survival and duration of response. When these patients were given this one drug without anything else, no other combination treatments, and these are patients that pretty much had everything else for treating their myeloma. So the average PFS, I don't have the number there, but it looks like it's around 9 or 10 months, and the duration of response here, again, about a year or so on median. I've got a patient on this who, she's out here somewhere, she's like 2 years out in complete remission still after a course of treatment that was limited, and then she stopped treatment and has been off treatment for a long time. her myeloma is completely in remission. So it definitely works well for some people and a good new treatment option. I think it's going to make it as a real drug for myeloma. So just a final few words about minimal residual disease in myeloma, because a lot of studies are using this as a marker of how well people are responding. But you can see that in the CAR T-cell studies, we're getting to the point that people are MRD negative but still relapsing. So just to understand that a little bit. So when we see myeloma under the microscope, we're kind of looking at the tip of the iceberg. We're looking at, you've got to think about myeloma on a log scale. One cell becomes 2 cells, becomes 4 cells, becomes 8 cells, becomes 16 cells. When we're looking at responses, we see, the last part of that cell division and the large number of tumor cells. But when we want to treat patients to a cure, we really want to get them all the way down to, every last single cell. And so beyond what we can see with a microscope or measure with protein in the blood, is a level of myeloma that we call minimal residual disease. So this is a level of myeloma where we can't detect anything by proteins or by looking down the microscope or even necessarily by x-rays or things like that. But we can only detect the myeloma perhaps by very sensitive methods called flow cytometry or deep sequencing of the bone marrow cells, looking for tumor cells that might be left behind there that we didn't see. And then when you get to the point where you can't detect any tumor cells by that point, by those investigations, then we start calling patients MRD negative. I mean, we can't detect any minimal residual disease. So that's kind of down here. And we're hoping that, if we get slightly deeper responses, we can get down to the point of cure where we've got rid of every single cell. But unfortunately, we're getting down to this point, but we're still seeing a lot of patients relapsing, often within a year from MRD negative if they're a late-stage myeloma patient. However, it is true that that MRD is very useful in predicting responses. So if we take all the bone marrow cells and we separate them out using different markers on the cell surface of those cells, we can identify small populations of plasma cells or myeloma cells. And depending on how many of those residual tumor cells there are in the bone marrow using these very sensitive techniques, we can start to predict how well patients are going to do. So if you've got less than 1 in a million, it's going to be a long time before the myeloma comes back. This is what we would call MRD negative. Whereas if you've got many more of those tumor cells still left behind, myeloma is going to come back quicker. In the CAR T-cell studies, what we're seeing though is that we get down to this level, but because these are patients that have been treated a lot, the myeloma does come back quicker than this. This is more of a newly diagnosed population that we're seeing really long responses with. So then the question comes, can we ever get down to the point where if we get really deep responses, can we cure myeloma? And there's not a lot of data on this yet because honestly we haven't been curing a lot of myeloma, but there is a little bit of data, so I thought I'd present that, finish off with a bit of a positive note. So in patients that were treated in the 1990s with plain old stem cell transplant, there was a fair rate of patients, 24%, that got a complete response to that kind of treatment, so about 1 in 4. And then of those patients, there were about 8% of patients who over the next 10 years or 12 years, they never showed signs of the myeloma coming back. And if we look at those patients, then those patients who stayed in complete response at 12 years post-transplant, if we followed them for another 5 or more years, there were no myeloma relapses happening. So this is what we call a plateau, and in other diseases, that's what we call a cure. So it looks like even from the 1990s, there was a small percentage of patients that were being cured, and we just really want to increase that number more and more. And I think we are probably with the improvements that we've made in the treatment of myeloma since. We just don't, you need to follow patients for so long to really know exactly where this number is now. So I could have put in about 1,000 slides here on my research that I do, which is really understanding why we're not curing myeloma and what we have to do to cure myeloma, because I think in the next decade we're going to cure myeloma. We just need to have that final understanding of what we're doing wrong. So anyway, I've reduced it down to 3 slides, so that shouldn't take too long. So this is what we typically think of our myeloma population here, plasma cells. They express this marker called CD38, which is what we target with when with daratumumab, and they express this marker called CD138, which kind of is similar to BCMA in its expression. And so the cells that are positive for both of those things are recognized as tumor cells, and that's what we measure for minimal residual disease. That's what we use to diagnose myeloma. Oops, sorry. And when we look at those cells under the microscope, we can see that they have these gene rearrangements here in yellow. This is a patient with something called translocation 414 myeloma, and you can see these translocation events that have happened inside this cell that make it a tumor cell. This patient also has an extra copy of chromosome 1, should only have 2 copies, it's got 3 copies. But if we look down at these cells here that are negative for 138 and sometimes negative for BCMA, we can see that within this population there are also myeloma cells, even though they're much smaller numbers here and it's really only a couple of % of these cells that are actually the tumor cells. We can find tumor cells down there that have the gain of chromosome 1q. And if we look at this really primitive population over here that's negative for both of those markers, they look completely different cells, but we can find that the myeloma genetic abnormalities are still inside those cells. And we don't target these cells with our current treatments, although we're just starting to. There are some groups that have, I think, taken on board our publication that there are myeloma cells that don't behave like the big population at the top right, that look different, and that they need to be targeted to really cure this myeloma. Because if we don't target these last few cells down here, then that's presumably where some of the relapses can occur from patients that have negative minimal residual disease and seem to have such great deep responses. So it's still a hypothesis, and we're working to prove that, but I honestly believe that's where we're failing to cure myeloma, and that we're not far off from curing it if we just broaden our targeting of the cells in myeloma. So there's another patient with hyperdiploid myeloma with some extra chromosomes. Should only have 2 of each of these colors in red and blue, and you can see all the way down to this population down here. We can break down other cells that actually are myeloma cells, even though they don't look like the typical plasma cell that we usually target with our treatments in myeloma. So this is like from 5 or 6 years ago. We published this story initially. So if we think about those plasma cells which we target with our current treatments, there are these petals on the flower, plasma cells. These are targeted by our standard treatments at present, like proteasome inhibitors, like Velcade, like even Daratumumab. But we have to start thinking about the seeds for those tumors and where the relapses are coming from. And we're not really doing a good job, I don't think, of targeting these particular cells, though there is progress in that area. The Chinese have really got on board with this, and they're doing a couple of CAR T-cell studies where they're trying to target the plasma cells, and they're trying to target B cells at the same time. And it'll be interesting to see how those CAR T-cell studies pan out. The results aren't reported yet, but there's a number of different strategies, I think, that we can employ to target all of these cells and get deeper responses and hopefully one day a cure. And I don't think we're that far away. I think we're really getting a lot closer. So what are we studying? That's not right. This is just to show, sorry, that last slide was a mistake for some reason. This is to show people working in my lab like on a Friday night at about 8 or 9 o'clock. You can see how dark it is outside through the windows. These are windows here. It's usually pretty bright out there. So some very dedicated people working to make progress in multiple myeloma. And some of our work is funded through the Myeloma Walk and other fundraising activities that you're all involved in. So thank you very much for that. So progress in multiple myeloma, I think we've covered a lot of topical areas, and a lot of this progress is due to your input in the research and participation. So thank you very much for those involved in clinical trials and fundraising. The question is, I don't have any hereditary cancer gene, but I still ended up getting myeloma, and why? And I tried to show that in that slide where there are these B cells in the bone marrow. They're there for fighting an infection. When they recognize an infection, they go into the lymph node and they shuffle all their genes. They cut up their DNA, their chromosomes, they rearrange their genes, and then they put it all back together. And hopefully that makes them better at fighting infection, so the next time you see the flu, you're not going to get the flu. That's how immunizations actually work. But when they do all that, it's a very unnatural thing to shuffle up your genes within the cell. they actually switch off some mechanisms that protect you from getting cancer so that they can do this to fight infection. It's a thing that only B cells do. They switch off the cancer protection mechanisms, they shuffle up their DNA, their genes to fight infection better, then they put it all back together, and then they try and switch on those anti-cancer genes. But occasionally they make mistakes. They shuffle the genes badly. It's just something that happens in one cell, in you, or someone else, or me, and then that one cell, over 10 or 20 years, starts to grow because it's shuffled up its genes the wrong way, and it now becomes a clone of daughter cells and sister cells and whatever, and they're all those cells are the same, and now you've got myeloma all of a sudden. And it was an effort of the body to fight an infection but it went wrong. Does early detection of multiple myeloma improve outcomes in terms of treatment? That's a great question, and people have argued over that a lot. I would say that in certain circumstances it can, and because of that, we've recently changed the IMWG, the International Myeloma Working Group, has recently changed the diagnosis a little bit of myeloma and allowed people to be diagnosed earlier. Some patients that we used to consider high-risk smoldering myeloma, we now consider active myeloma, and we start treatment, whereas previously we would have continued to watch those patients. And the reason is there was 1 or 2 studies that have suggested that there probably is a benefit from treating those patients earlier if you use the right treatment. In the olden days, we used treatments that weren't that good, and there was really no benefit from treating earlier. Right, it's a really a difficult question because it comes down to finances of screening programs, which there's a lot of cancers that we can screen for, and really what needs to be worked out is there a benefit for doing that screening relative to the cost instead of taking the money that might use to fund Daratumumab and putting it into screening, for example. And so a lot of governments employ people with, employ doctors and health economists to try and work those kind of questions out. You're right, if you go do the serum protein electrophoresis or SPEP study, you can find a lot more patients with a monoclonal protein in the blood. And like I said, there's 3% of people over the age of 50, 5% of people over the age of 70 are going to have that. Should we be trying to identify all of those patients and then following them all and starting treatment earlier on some of them? And at the moment, the benefits of starting treatment early are relatively modest for most people, but for some people it could be really important. But you're going to identify a lot of patients that are going to need to be followed for a long time. It's going to cost a lot of money, and is that money better spent doing that than versus paying for Daratumumab when you actually get diagnosed? I don't know the answer, but I'd be supportive of screening more people and knowing earlier about the monoclonal proteins. Yeah. For newly diagnosed patients receiving CyBOR D, should we add Daratumumab now? The truth is we don't really have the answer because it hasn't been studied together with CyBOR D Frontline, although it has been studied with VMP Frontline, which is a similar regimen to CyBOR D, and it did well there. It's not funded by the government, at least not in Ontario, for frontline use at the moment. So we can't add it. I can't go and prescribe it for any of my patients who are on Revlimid and dexamethasone or on VMP or on CyBOR D Frontline. It's not funded. But if you have private insurance, maybe you can make a case when we now have data that this works well in frontline patients and I want it added, and maybe you should fund that for me, please. Yeah, so I guess the question is, if you use Daratumumab early on, will it work as well again later on, or have you really lost the benefit of using it later on? Yeah. You probably have lost the benefit of using it later on, but you've gained it up front early on, and it's probably going to be a bigger benefit early on than later on. So you're still better off using it earlier rather than later. Yeah. And at the moment, there's no one that's done any studies, I think, with reusing Daratumumab or trying to use it again later on, so we don't really know. So if someone's going to pay for genomic sequencing of myeloma, can that be used for managing newly diagnosed myeloma patients?
So would that change the regimen?
Yeah. So it's another good question. A lot of groups are looking at profiling myeloma in a very detailed way, looking at the genes and mutations that make that particular tumor happen in that patient versus another myeloma in another patient, and trying to personalize the treatment to your tumor, a different treatment. Unfortunately, the number of drugs that really work for myeloma is pretty limited, and mostly we don't know exactly what causes the resistance to those drugs very well. And so it's hard to predict who's going to respond to this drug or that drug, and yet we have all these clinical trials that say this regimen works best when we give it to this bunch of people versus that one when we give it to that bunch of people. So we end up making these decisions based on population-based studies. We really need a lot more information to be able to come up with a personalized treatment up front, but people are working on it. Right. So the question is, there's a lot of buzz in the US about personalized medicine. They're doing this in a lot of different cancers. Is this because they've got more drugs to use? Are they ahead of us? There is a lot of buzz about personalized medicine. I'm taking a slightly one step back from that buzz. I think people who are researching in this area, they are very enthusiastic about it. So yes, if you happen to find a mutation in one particular pathway, there is often a drug for that pathway, and if you put all the drugs together, that you can target different pathways in cancer cells. There's a lot of those drugs out there, and in Canada too. In myeloma, it's less numbers of pathways targeted than in other cancers, but nevertheless, there may be drugs that work in myeloma that we didn't expect was going to work in myeloma, but because this patient has this particular mutation in this particular pathway, they might respond to that drug. But the downside is then you're taking a patient and you're giving them one drug that often is going to work on some of their cells, but that mutation is not going to be in all their myeloma cells, and the cells that don't have it is going to grow back. And we find that when it's been done in myeloma, the other cells grow back in a few months. And so you've spent all that effort trying to come up with the exact drug that's going to work, but it really only worked for a few months, and now you're back to square one. And so there is potential for doing it, but patients are probably then going to need to be on combinations of drugs to target multiple pathways, and it ends up being chemotherapy just like any other treatment that we give, not necessarily any better. I really like the immune therapy approach where we're targeting markers on the outside of the cells that just about all patients have. We can go in there, we can target all of those patients with the same drug. Those markers are on all patient cells. We're just not getting all of the right cells, I don't think. I think we need to cover a bigger spectrum of cells to really eradicate the myeloma clone and get these deeper responses. Right, so the question is the Daratumumab only for those who had stem cell transplant? And some of that data that I presented was for patients who didn't have stem cell transplant. So no, actually patients with relapsed myeloma, irrespective of whether you had transplant or not, can benefit from this. Patients who aren't having a transplant, there's a lot of data that it works well up front now. So it's patients who aren't having transplant that we show Frontline that the Daratumumab works well. For patients who are going on to have a transplant as part of their treatment, there are studies using Daratumumab in the frontline setting, but because they tend to have longer remissions anyway after the transplant, we're still waiting for the results to tell us whether adding Daratumumab up front is useful or not. But it probably will be, but we're waiting to see. So the question is, what can you do, I think, to delay smoldering myeloma from progressing? Is vitamins or omega doses of things going to help? We don't have any real strong data for any of that making an improvement in terms of smoldering myeloma progressing or staying stable. What I would say is we have some evidence that smoldering myeloma is held at bay by your immune system. So there are a few models of smoldering myeloma in the research fields that suggest that your immune system is pretty important in keeping it suppressed and not allowing it to progress. So really you want to do everything you can to keep yourself healthy and your immune system at its best. And I think that just means eating a healthy diet and getting a bit of exercise and doing all the usual things just to stay as healthy as you can. Yep. So most insurance companies are going to want evidence that it's an effective treatment that they're going to pay for. And so they're going to want some sort of clinical trial that shows paying for the Daratumumab up front before a stem cell transplant is a good thing to do, and that it's going to make you healthier and they should pay for it.
You have to make a case, right?
You have to make a case, and so it depends on the quality of data that they need, but a lot of insurance companies are going to want phase 3 randomized data showing that this is something that works better than not giving the Daratumumab up front.
You have it in phase 3.
Not for transplant eligible, only for the transplant ineligible patients so far, but the other studies are coming, but they're going to take a bit longer to